Hello everyone, today's Lunch and Learn topic is Uncover Spatial Molecular Dynamics with Area Scan Detector. As we all know, we can do a lot of experiments on confocal systems. The sample can scaling from 2D cell cultures to new 3D model systems, and confocal system can offer us great performance on sensitivity, resolution, speed, spectral flexibility. But confocal system can offer us to measure the molecular dynamics as well, to give us an molecular rearrangement. The most popular way of measure molecular dynamics is by using the method of FRAP, fluorescent recovery after photobleaching. This is a method by bleaching one region of the cell and to record the fluorescent recovery of the region of the cell. This method is very accessible and can work with many samples, but it also has some compromise, like you have to bleach your sample, so high laser power is needed, which can perturb the sample. And most of the case, you can only do one experiment per cell. Secondly, the information is averaged over multiple populations and compartments. I can remember people usually ask me, can I do FRAP on a very, very small region? Like I can draw a very small circle to do the bleach, which is kind of difficult. So actually on confocal system, there is another option to measure molecular dynamics, which is FCS, fluorescence correlation spectroscopy. This is a method by assessing molecular dynamics by measuring the fluorescence going in and going out of the observation volume, and the movement will be recorded by the count rate, and the count rate curve will be mathematically extract a lot of information on different parameters like molecular concentration, mobility, interactions, identification of subpopulations. This FCS technology can adapt for a wide range of questions, but it also have some drawbacks. For example, it only works on very dim samples, and most of the time, the high training effort is needed, and specific hardware is needed. Let's have a closer look at what is FCS. Instead of scanning in FCS, we park our excitation laser at certain spots and observe the fluorescent molecules passing through this spot. This will create some intensity fluctuations. Normally, we will record intensity curve, and by using this intensity trace, we will do the autocorrelation. This is purely math, and we will create a correlation curve. Let's imagine the intensity traces look like this. The autocorrelation is we duplicate this intensity trace and compare it with itself by shifting in time. So at the beginning, the similarity is very high, but when we're shifting in time, the similarity will become less and less. At a certain time point, it will drop into zero. From this correlation curve, we already can get a lot of information. For example, we can get the information of number of molecules from the amplitude of the correlation curve. If we have higher concentration, the amplitude of the correlation curve is lower. if we normalize the amplitude of the correlation curve, we will eliminate the information of the number of molecules, but we will get the information of diffusion speed. If we have faster diffusion speed, the correlation curve will drop into zero faster. So from this curve, we can get the information of the diffusion speed. If we want to get more information from the correlation curve, we need a couple of more things, like we need to know the PSF volume. If we know the number of molecules, we know the PSF volume, we can calculate the concentration. And secondly, we need to fit our correlation curve into a model that theoretically best describe the molecular movement. If we get a better model, we can get a better result. So for our life scientists, the FCS technology sometimes seems a little bit intimidating because it involves a lot of model fitting, curves, and statistics. But this is not the main reason why there are not too many people use FCS technology in our life science research, because FCS technology only can measure very, very dim sample. Like there are 4 cells in this region, only this very dim cell can be measured by FCS. These 2 bright cells, which is more commonly exist in our experiment, is impossible to do FCS. At the other end, this paper, this overall paper shows us actually FCS is a good tool to revealing hidden information of the molecular dynamics. Like FCS can provide us information of molecular concentration. We can get the information of the molecular concentration at a certain spot in the cell when the protein conduct certain kind of functions. This is very important message for us to understand our protein or our biological process. And we can get the information of mobility. We can get the information of how the molecule is transported directly or it's transported through membrane binding or has some hindrance, and we can get the information about the molecular size, like its monomer or dimer or oligomer during its functioning. So Zeiss started to adapt the FCS technology from 1996, from the comfort core to 780 to 880 to 980. Especially at the year of 2018, the new technology of use spatial information of error scan detector in the FCS technology has published. Zeiss started to rethink FCS. We not only want to use spatial information by error scan detector, but we also want to increase the usability. We want FCS can measure all samples and guaranteed right hardware settings. Wanted to have the quality control of the measurement, orientation within the cell, and less complexity. So we launched the new function of the Dynamics Profiler. As the dynamic profiler is using error scan detector to do the FCS. I want to recap the error scan detector a little bit. Instead of using one GASPA or one PMT as a detector to collect the signal, error scan detector is using 32 GASPA-based detector element array to collect the signal. As I mentioned, the traditional FCS can only work for dim samples, so only these two very dim cell can do FCS. But in dynamic profilers, you can do FCS on all the samples. Even this two bright cell can do FCS in dynamic profiler. Why? Because firstly, we use the analog mode of the detector, so it capitalizes the huge dynamic range and it transferred to the equivalent photo counting. And secondly, it used a sum value of 19 individual center detectors. So for each detector element, it only sees a fraction of the signal. So it also increases the dynamic range. So in this method, error scan detector is used as a single detector element. The sum of the 19 detector elements is record the intensity trace of the molecules passing through this observation volume. Let's imagine there is only one fluorescent molecules passing through this observation volume. The sum of the 19 detector elements record the intensity trace. And then we will duplicate this intensity trace and compare with itself by shifting in time, and then we get the correlation curve. And from this correlation curve, we can already get a lot of information, like we can get the information of mobility and concentration. Let's see some example. So again, these 4 cells, as we mentioned in the FCS, like in the past, we only can measure these 2 cells. But now, if we put 4 spots, 4 measurement spots on 4 different cells, we can get the correlation curve of all the samples, and from the dynamic profilers, we can get the concentration, and we can measure the number of molecules on both dim cell and bright cell. Another example of the bright sample, dynamic profiler can measure the bright sample. This is the E. coli express GFP. It is a very bright sample. In the past, it's impossible to do FCS, but in dynamic profiler, you can get the correlation curve, and you can get the data of the diffusion coefficient, concentration directly in the dynamic profiler. This is the third example. This one is a U2 OS cell, and the magenta is protein fiber layering. People want to understand the molecular dynamics of the fiber layering in the nucleolus and in the nucleoplasma. So although the fiber layering in the nucleolus is very bright, but we can get the correlation curve in the dynamic profiler of 4 different spots. And if we normalize the amplitude of the correlation curve, we can get the difference of the diffusion speed in the nucleolus and in the nucleoplasma. This example is Drosophila embryo, and the red is the M. cherry. The expression level is controlled by the cardiac enhancer, and we can see in different cells the expression level, the brightness is different. Some samples, in some cell it's very dim, but in some cell it's very bright. In dynamic profiler, we can get the correlation curve at all different cells, and if we normalize the amplitude of the correlation curve, we can get the information, which is the diffusion speed of every place is same. And we go to another place. We measure 5 different cells that have different brightness, but if we normalize the amplitude of the correlation curve, we can get the information of the diffusion speed. As we talk about in dynamic profiler, we can measure all the samples, even for the bright sample. But how about the usability? So the interface of the dynamic profiler is wizard, so it's very easy, user-friendly. Dynamic profiler is accessible only with the correct hardware chosen, and it will include a fast, automatic, precise error scan alignment, so you don't need to worry about the PSF alignment in the FCS, and it ensures all necessary steps to be done and has the quality control of the measurement, which is the CPM, the color of the CPM, and it has a reference image to let you to control the sample movement. So this is the interface of the dynamic profiler. You can have a reference image, and I want to especially mention this is the circle of the measurement spot is representing exact space error scan detector measured, and we have a easy to read table, and the color of the CPM is the quality control, and we get a correlation curve just directly on the interface of the software. And as we mentioned, we need to do the fitting after you get the correlation curve. In dynamic profiler, you don't need to do the model fitting by yourself. Mathematically, we include the most common one, so people normally use it for most of the experiment, so you just need to choose which one is suitable for you. Except for those conventional results we can get from the FCS, dynamic profiler offer us 2 more analysis options. The first one is flow analysis. Dynamic profiler can use spatial information of the error scan detector to give us the information of the flow speed and flow direction of certain molecules at certain spots. So this information is very important for some studies of the active movement of solution, like bloodstreams, or applications in the microfluid systems. So in this, error scan detector does not work used as a single detector. We now use it in a different way. Like we use the two detector element as a pair and correlate them with each other. This is called cross correlation. And from this cross correlation, we can know if we can see molecules passing through one detector element, whether we can see it passing through another detector element a little bit later. Let's have a closer look at this cross correlation in the flow analysis. We now use the two detector element as a pair and correlate them with each other. It means the intensity trace of one detector element and the intensity trace of another detector element will be time compared. And because the molecules needs time to travel from one detector element to another detector element, so this cross correlation will exhibit a time delay. We will do this cross correlation for one pair on both directions, but we will know only in the flow direction we can have this time delayed cross correlation. And we will do this cross correlation along 3 axes, and each axis we will have 9 pairs. But you don't need to worry about all this calculation and flow angles. In the interface of the dynamic profiler, you can directly get the result of the flow direction and flow speed. Let's dive into one example. This is a fluorescent labeled duction injected into the blood vessel of zebrafish larva. People want to understand the flow speed and flow direction on certain kind of small, exactly spot of the duction. So this image is an overview of the zebrafish larva captured by the low magnificent lens. Then we zoom in to certain spots that we want to study. We can place some measurement spots, and in the dynamic profiler, you can get the exact flow speed and flow direction of the duction. Another application is for the organ-on-a-chip technology, which is widely used for people to understand human disease and treatment. So inside of the microfluid chamber, it usually it's a very complex environment, contains many layers, many cells, and definitely people want to understand the flow speed and flow direction of certain drugs applied to certain place of the cell. Dynamic profiler can offer some options to measure that. For example, we can fill the microfluid chamber with rhodamine and place the measurement spot from the border to the center of the channel, and we can get the exact flow speed at certain spot. We can see at the border the flow speed is relatively slow. At the center, the flow speed is relatively high, and you can draw the flow profile along the channel. And you can also do this same thing along z-axis, and you can put different measurement spots on different z to get the flow profile along z. The third analysis option is to measure the asymmetric diffusion behavior. So you can measure the different molecular behavior in cell compartment, barriers, and condensates. In this measurement, air scan detector works in a different way. The center detector element is correlates with the third ring of the detector element. So along each axis, we will have a cross correlation curve, and these cross correlation curve can be compared with each other by in regard to number of molecules and diffusion speed. We will use color-coded heat map to visually representing the relative height and relative length. If we focus on the concentration difference, so if at certain axis there are more red, means the molecule behaves like at certain axis have slow concentration. If we focus on diffusion speed differences, if at certain axis has more red, means the molecules behaves at certain axis have slower diffusion speed. So in this method, we can visualize the differences in molecular behavior without time delay within one single PSF along 6 different axes. There is one application is by study the biomolecular condensates. From this overview paper, we can see there are many kinds of condensates exist in our cell, and they actively participate the biological reactions, but they are formed by the lipid-lipid phase separation, so they don't have a membrane structure around them. So people really want to study the asymmetric molecular behaviors around the border of the condensates. Let's dive into one example. This is the HeLa cell. After the heat shock, it will form the GFP-labeled stress granules. Those stress granules formed by lipid-lipid phase separation, so they don't have a membrane to enclose them. So if we zoom in to study one of the stress granules, we put some measurement spots, and from the dynamic profiler, you can get the asymmetric diffusion measurement result. So from this border, we can see along these axes, the molecule behaves slower diffusion compared with the other directions. But in the center of the stress granules, the molecules behaves like have unified diffusion speed. On the other border, it shows the same pattern. We can not only study the condensates, but we also can study some other examples like barriers and compartments. Like this one is a lipid bilayer contains nanomolar concentration of far-red fluorescent dye. And if we put the measurement spot in the center, we can get the correlation curve. And from the dynamic profiler, after fitting, we can get the concentration and diffusion coefficient. And in the asymmetric diffusion measurement, you can study the different molecule behavior at different positions. So in summary, in Dynamic Profiler, one recording you can have 3 powerful analysis. You can get the information of molecular concentration and mobility. You can get the information of asymmetric diffusion measurement, like the different molecular behaviors in cell compartment barriers and condensates. You can get the flow speed and flow direction of certain molecules at certain spot. So by using error scan detector, you not only can get the super resolution high-speed imaging. Now you can get the you can measure the molecular dynamics. Last but the most important, I want to mention, in HCBI main campus, you can choose LSM 980 to do the molecular dynamic profiler. And remember to choose the FCS objective lens. And at the lab at Austin, you can choose the LSM 900 Bio to do the dynamic profiler. There is a 40x water objective. And if you want to get more information, you can scan this QR code. Thank you very much.